It is my great pleasure to welcome today Julijana Ivanisevic, who will lead a conference entitled Metabolic View of Sex Hormone Status and Health Risk, a longitudinal metabolomy-wide association study. Dr. Ivanisevic is head of the Metabolomics Lipidomics platform, senior lecturer at the Faculty of Biology at Lausanne. She joined the University of Lausanne in 2015, following a postdoctoral training at the Center for Metabolomics and Mass Spectrometry at the Scripps Research Institute in Napa, California. This is a world-famous institute in this field, and it's really a lot of great research is being conducted there. And so she has a PhD in chemical biology in France. And so she really has this extremely precious expertise that is being now implemented using biosamples from cohorts, in particular population-based cohorts. It's a great pleasure to welcome you today, and give you the floor. Thank you.
Thank you. So hello, everyone. Thank you very much, Professor Gorschup, for your invitation, and yes, for this very nice introduction. I'm really pleased to actually contribute to this seminar series of your department with this presentation of recent work we have done on metabolic phenotyping of apparently healthy populations. So we have selected a subgroup of participants, of several thousand participants within Colloque. And we also work, we are involved in the metabolic phenotyping of a complete health cohort, which has been established by the sports medicine team at the University of Lausanne. And so prior to really showing you some of these preliminary results we managed to acquire on these population studies, I will first try to provide you an overview on metabolomics, including the approaches we are using, the challenges we are still facing, and then also the advantages really of studying the metabolome. And then I will present you briefly the results we have obtained until now, during this past year and a half, when we started, really, since we started to work on complete health and subgroup of Colloque. So due to very recent advancements, actually, but significant advancements in the sensitivity and the specificity of metabolic measurements, we can actually say that today we are in a way revisiting or rethinking the metabolism with metabolomics and actually building upon this acquired knowledge in biochemistry during this era of biochemical genetics. Why so? Because I have the impression that during this era, we have actually, the metabolism research has focused much more on the genes and their function. And today, in this post-genomics era of biochemistry, we are focusing back on metabolites and their role, like really in the very beginnings, or actually, of the biochemical understanding of metabolism. And this comeback was mainly, is mainly a result really of very significant advancements, as I said, mainly in technology, and this specifically mass spectrometry technology together with NMR, though. Then as well, the computing power and the bioinformatics solutions, the computational solutions that actually allow us to process and to analyze these extremely big and complex data sets. And this is why metabolomics is often qualified today as a next generation metabolic profiling that allows us to measure in a very comprehensive way, not only these highly abundant nutrients, so amino acids and energy currency metabolites that are actually involved directly in energy production and storage, so the energy flow through metabolism, but also these very low abundant messenger molecules that actually really mediate the information flow through chemical signaling. And here I really love to cite the Professor David Wishart, who is one of the pioneers in metabolomics field, who said that while the metabolites may be small, and we indeed treat them as small molecules, they are small in their mass, so below 1,500 Daltons, their influence actually in human physiology and disease is truly profound. And there are multiple examples and studies that show today that metabolites are perhaps the body's really most important signaling molecules. And this is justified, as I said, by these multiple studies that have recently actually demonstrated that we should not keep considering metabolites only as the building blocks of structural components of cells or fuels for cellular genetics, but that we should really start perceiving them as really active modulators of gene expression and of protein activity, and therefore also multiple biological processes, including all of these that are illustrated actually on this slide, such as the cell differentiation, cell activation, cell proliferation, survival and death, so the apoptosis. And there are really multiple papers that actually nicely show the examples where very common metabolites that we thought that we had this common idea about them as fuels, like for example succinate, are actually capable of really modulating significantly different biological processes, and also our phenotyping different model systems that we are studying. And in line with these findings, the multiple studies have also shown that metabolites play a central role in multiple pathophysiological processes. And here I try to also give you some examples by these recent reviews. So we do know that metabolites can harm and that they can kill, and that they can actually have oncogenic, diabetogenic, and atherogenic properties, especially if they accumulate over longer periods of time. So if we talk about the chronic accumulation. And to illustrate this, I wanted to show you several examples that I think, that today are really well established and have already been validated in multiple human cohorts. So one of them is the TMAO, or the 3-methylamine oxide, that we do know has important atherogenic properties. It has been really associated with the atherosclerosis. It is known to promote actually the development of plaque in vascular system. And what is quite important is that TMAO, actually its source is really the fatty food and meat mainly. And it's extremely important to consider that the gut microbiome, so the intestinal microbiome, will here play a very important role in the synthesis, in the production of TMAO. We can go in more details in the mechanism later on if this interests you, but for the sake of time, I won't enter in many more details. But we do know that its accumulation will actually promote the formation or the conversion of macrophages to the foam cells that will then actually lead to the development of plaques in the vascular system. Yet another great example are the branched chain amino acids, which have been clearly associated as well with the significant risk actually of developing obesity, type 2 diabetes, as well as different cardiovascular diseases. So they are known to activate the mTOR signaling cascade, and in this way actually stimulate the proliferation, the growth of beta cells, and the production of insulin. So in the way they mimic this state of the chronical exposure, chronically high insulin levels, which will finally lead to the insulin resistance. Again, the mechanism has been described in many details, but it's also a continuous debate, because we are constantly discovering new pathways actually, and new signaling pathways actually in different model organisms. Yet another great example are ceramides. So ceramides that are actually a part of the Sphingolipid class, so a specific class of lipids that have also been associated really with the insulin resistance, with atherosclerosis, and we do know that their accumulation is actually driven by inflammation and by the excessive calorie supply. And so based on all of these examples, actually, we can clearly say that today, using different metabolomic approaches, we are really rewriting the disease etiology. And we are constantly discovering new things about the metabolite role, I mean, the role that metabolites actually play in the onset and the development of different metabolic diseases. So all these discoveries I have presented have been initiated by metabolomics approaches, and we were able to actually decipher the role of these metabolites using different metabolomic approaches. I mean, combining them, of course, with different other experimental orthogonal approaches. And so we can say actually that metabolomics allow us not only to probe the biochemistry at the cellular level, but also at the more systemic organism level. And it allows us not only to measure the small molecules from the body, so these gene-derived endogenous metabolites, but also the environmentally derived metabolites. And in this way, we actually use the metabolic profiles to identify these different environmental exposures, so including our dietary regimen, the level of our physical activity, and all the other exposures to pollutants or toxicants. And in this way, we can actually help us in the way the metabolic profiles actually help us to decipher to which level actually these different environmental exposures will contribute to the onset and development of these complex cardiometabolic diseases. And this is why, because these metabolic profiles actually do reflect or are a product of gene and protein activity on one side, so reflect really the gene function, and on the other side also our lifestyle and different types of exposure, as I just said, they are often classified as an intermediary phenotype at the molecular level. And that provide us a very close link actually to these endpoint phenotypes or clinical, the health outcomes. But what is really important here is that these metabolic profiles or phenotypes are genetically much less complex, so less polygenic compared to these endpoint phenotypes, but still equally heritable. And this is why actually these metabolic levels can be instrumented successfully by the robustly associated independent genetic variants to, at the end, test really their causal effect on cardiometabolic risk factors. And this is what I will show you a few results that we were able to acquire in collaboration with Professor Kutalik's team on the Colloque population. And so here on this scheme, I just wanted to illustrate you to which point, and today it's really important to combine these standardized measurements of different clinical parameters, such as the cardiorespiratory fitness or the routine blood analysis, with the metabolic profiles or the metabolomic markers that will actually capture the response to challenges that are due to our lifestyle. And this is not only to really improve the discrimination of risk for development of different diseases, but also to identify patients' behaviors that are amenable to changes, and hopefully early enough. So the main idea actually by implementing in a way this omic scale metabolic profiling in the clinical practice would really be to significantly improve the health monitoring, and specifically the healthy life expectancy, so the health span, and not just the lifespan by different types of treatments and medication. And because these very complex metabolic profiles capture actually the metabolic and or biochemical individuality, if you want, they are really today seen, or they are emerging as a really very powerful phenotyping tool to make us move towards this more precise approach to medical care. And here it's really important indeed to evaluate, to explore further and to evaluate the predictive capacity of these signatures that would then allow us really to identify the individuals that are at risk of developing different metabolic diseases, but early enough. Here I'm speaking about tens of years before, so 10 to 30 years before. And there are many studies that nicely show this. Show this capacity that is indeed inherent to these signatures. So here I wanted to show you actually on this slide into how many details, how deep actually we can go with the measurement of the metabolome. So as I said previously, the technology has advanced significantly, and we can today measure the metabolites with much more sensitivity and specificity. And this allows us to go down to the molecular species level. And here I wanted to share with you one profile of the sphingolipid metabolism that we are now performing, acquiring on a routine basis at our platform. This profile, as you can see, englobe many different species of sphingolipids, so including different ceramides, dihydroceramides, hexosylceramides, sphingomyelins, etc. So overall, we are able to acquire a profile which englobes more than 60 different species. And so you may ask yourself, why is it so important to go down to really the molecular species level? Well, I would say mainly because it is today known that the structurally very closely related species can have a distinct biological function and be associated with distinct metabolic consequences. And here I wanted to show you this on one example of two ceramides, where you will see that one of them has one unsaturation more compared to the other one. And this specific ceramide species, together with some others, has been associated and positively associated with aging, with the risk of atherosclerosis, and in addition, later on, with many adverse, really clinical consequences. So just keep this in mind that indeed it is species specific. And we cannot anymore rely just on the measurement of the total lipid content. We can go much further. The technology has evolved. And not only the technology, also the precision of the measurements that I will show you later on. And so these ceramides, actually, and in specific species of ceramides, have indeed been revealed that in the same way as cholesterol, will really accumulate in atherosclerotic lesions, and they have, as I previously said, have been really associated with many different health outcomes, including different cardiovascular diseases. Their mechanism of action is, though, different than the mechanism of cholesterol and is independent on the cholesterol metabolism. And here, this is something I wanted to share with you because it has already been implemented in clinical practice in US, specifically in Mayo Clinic. So they have actually developed the clinical score based on these 4 specific ceramide species. And this score, when combined with the really traditional score that is based on the blood pressure, total cholesterol. So when these 2 scores are combined, they have actually noticed that the accuracy and the sensitivity of the prediction models is much higher compared just to the traditional score based on this, let's say, standardized clinical measurements. And so this is one great example that actually shows how metabolomics and these recent discoveries in metabolomics can actually slowly be transferred to the clinical practice. The problem is, of course, that we still know much less about the ceramide metabolism than we know about cholesterol metabolism, but there is really a debate in the field whether very soon actually the ceramides will replace cholesterol and will really be used as markers that are much more sensitive actually and much more accurate for the prognosis, but also the diagnosis of different cardiovascular disorders. So yeah, I just wanted to share with you this example because it's actually already implemented in clinical practice, but we are still there is still a lot of debate because we are still exploring the mechanism of their action. We still have to get more knowledge about really, more mechanistic findings, also to be able to then advise people about the potential treatment, or I don't know, more, subscribe them, advise them about higher physical activity or to change their dietary regimens, etc. So this example is just one of the examples that nicely shows how this really structural and functional diversity of metabolites actually represents a very, very rich resource to hopefully improve really the cardiometabolic risk assessment and at the end, I mean, more precise, I would say, rather health monitoring. And despite of the fact that I have highlighted many times that yes, indeed, the specificity and the sensitivity of the metabolic measurement has enhanced significantly, but we are still facing many challenges in the field. And as you can see, these challenges are really related to the metabolic diversity and the size of the metabolome. So you can see that compared to the proteins and genes, the metabolites are extremely, I mean, kind of harbor really an extreme chemical diversity. There are really no even limits on how they can be assembled. And we today estimate that their number is close to 1 million different species. I mean, new species and especially new lipid species are being discovered almost on a daily basis. And on the top of this really, let's say, structural heterogeneity, metabolites are also present in an extremely wide concentration ranges that span more than 11 orders of magnitude in a complex biological matrix such as plasma. And due to this, we have this also technological challenge to really measure simultaneously these metabolites that are present in a very, very low concentrations and those that are actually present in very high concentrations, such as cholesterol, for example. And here I just wanted to illustrate this issue on the composition of the human plasma lipidome, where you can see actually that just only the lipidome in human plasma will span more than 9 orders of magnitude. So starting from these oxylipins that are really mediators of inflammatory processes that are present in the low picomolar levels and going up to the cholesterol esters, fatty acids, many different phospholipids, glycerolipids that are present in much higher levels, so up to millimolar levels. So you can just imagine that when you have to measure this entire range, your instrument, you have to have the adaptive technology to actually englobe all this very wide range of concentrations in which metabolites are present in different biological matrices. And so, although indeed today we can measure hundreds of polar and thousands even of different lipid species from only very limited amounts of biological samples, still there is no universal technique simply because one size does not fit all this metabolite diversity. And for this reason, we often use 2 different approaches. So the targeted approach with which we typically focus on one pathway or one class of metabolites of interest, and on the other extreme, the untargeted approach with which we are trying to detect as many metabolites as possible. So I must say that the limit actually between these 2 approaches is becoming narrower and narrower, and many labs actually are developing what we call today high coverage targeted methods. Why? Because they serve us as an alternative to untargeted approaches, because with this type of methods, we can indeed measure in a reliable way several hundred known polar and thousands of different lipid species. And we have also installed and established, set up these methods in our lab. So in a way, these methods serve us as an alternative for untargeted, because of their high coverage, they still allow us to actually investigate the metabolism really in depth. So to perform this really deep annotation of the chemical composition of different matrices. And here I can show you this just on the example of human plasma and the methods we have set up in our lab. So from human plasma, from only 5 microliters of human plasma, we can measure more than 200 different polar metabolites and more than 700 different complex lipid species. And what is important here, please consider these are not just the detectable metabolites, these are measurable metabolites. So when I say measurable, this means with a really high precision, so the precision that is indeed prescribed by the FDA. So we are, in terms of the interday precision, the coefficients of variations are from 2 to 20% at most for all of these 700 complex lipid species. And what has really allowed us to, in a way, apply this type of methodologies to this large-scale population studies is the fact that we have tried to automate as best as possible the sample preparation. Why? To make it to actually maximize the reproducibility of the sample preparation, because we have noticed that this is where the most of the bias is actually introduced during the manual sample preparation. Then we, of course, developed this, let's say standardized the analysis in the way that they are as high throughput as possible. So you will see that in 12 minutes, which is a total analysis time per sample, we can indeed measure all of these 700 lipid species in human plasma. And on the top of this, we are, with this type of targeted approaches, we are actually reporting, acquiring the quantitative data. In my field, quantitative means the absolute concentrations, meaning that these concentrations have been normalized. So we do use the internal standard spike, and we do account for actually the extraction efficiency. We do normalize for matrix effects as well. Because believe me, the plasma from a person who has a disease will have different matrix effect from the plasma of a person who is considered healthy. Simply because the lipid composition will be very different, and these lipids that might be present in a very high concentration can mask actually the signal. And in this way can suppress the signal, and the fold change that, for example, you will get out of the untargeted approaches can be significantly biased by these matrix effects and can actually be just a false or negative results. So this is just to,
I would have a question regarding what you just said. Is it a problem for you if you are handling non-fasting samples? But if somebody has eaten a fondue just 1 hour before, would it be a problem for this?
Yes. Yes, because for sure we know that the diet is the source of many lipids. So for sure, yes, the composition of plasma will be significantly different in those people compared to.
But do you prefer having only fasting samples?
Yeah, a priori, this is what has been done until now, or at least we apply a certain number of hours of fasting after a meal. This is what we have done with the sports medicine doctors. At least it was mandatory to have fasted for 3 hours prior to the blood collection. But yes, this is indeed this is an issue. Everything depends what you are studying and how well you can control for the diet as well. But in humans. Unless we are kept in controlled conditions. And so here, yes, I wanted to really highlight this question because many times we get this question. Should people come to us, want to perform metabolomics and then have this idea about untargeted metabolomics and tell us, yes, I really want to perform untargeted analysis because I have heard that if we do untargeted, then we will be able to actually profile everything that is present in a sample. This is not true. Please don't believe them. This approach was extremely trendy for the past, several decades, simply because in this way we were able to say that yes, we as well can do omic scale analysis, but we are really far from that still. Main challenges you have seen, it's really metabolite diversity. So what happens actually when you do use untargeted assays, they can be considered as unbiased only because you do not have an a priori hypothesis. But other than that, they are heavily biased. And they are heavily biased mainly by the lack of sensitivity in this full scan mode. And so this is what I really wanted to illustrate by this mountain analogy. So the coverage that you will actually get with these untargeted assays will be limited to this well-trodden foothills in a way that, and in most of the cases, what you will be able to measure with a targeted approach are these very well-known, highly abundant nutrients. You always have the potential to discover something unknown if it ionizes very well, if it's highly abundant. So it's possible to potentially identify some unknowns, some xenobiotics that will, for example, ionize very well. But just keep this in mind. It makes sense to apply untargeted approaches when you are studying, for example, the drug metabolism, and you have to identify new metabolites of a specific drug, or when you study natural products. But if you just want to really quantitatively profile known metabolites in the context of clinical population studies, I really do not believe this is the best way to go for many different reasons. And on the other hand, if you look at the coverage of targeted assays, so yes, for sure, they are limited to knowns because we have to actually provide to the instrument the list of known metabolites we will target. Still, their coverage spans the entire range of concentrations. So with the targeted assays, we can indeed measure these very low abundant signaling molecules that can be extremely important in some physiological processes. So yes, please consider, have this in mind, but I mean, we can discuss this in more details. And on the top of this, like I said previously, targeted assays have this potential, have this advantage of being quantitative. And in human populations, it's extremely important. Why? Because the inter-individual variability due to our different lifestyles is extremely high. And on the other side, the changes that we are able to actually reveal or to identify are very small. So the changes that we see span from 20 to 60% at most between the control and the person with and let's say confirmed disease, which then these changes are subtle, they are physiologically relevant, but if you just use untargeted approaches, your chances to really discover them and to quantify them are much, much lower than if you really use targeted approach, so with the internal standard spike and then the data normalization that goes with it. And on the top of this, when we do acquire the quantitative data, this also allows you to make the cross-laboratory or cross-study comparisons, actually, to compare the data that were acquired in the context of different population studies by different labs and potentially different methods and technologies. What is also very important is that all these approaches, no matter if they're targeted, so single pathway, multiple pathway targeted or untargeted, can be applied from different model systems to really large-scale population studies. And this is mainly due to this high throughput of metabolomic, of metabolomic approaches. So we are really on a daily basis profiling, for example, different fractions enriched in different organelles, such as lipid droplets, mitochondria, endoplasmatic reticulum, etc. We do profile organoids, the spheres, different cell lines, tissues coming from many different model organisms, and then, of course, also the samples, so biofluids or the tissue biopsies come from, collected from humans. So this is just for you to be aware of this, because some people do not really know that indeed all these different, this very diverse sample panel can be analyzed. And now I will present you some of these preliminary results that we have acquired in the context of complete health and the selected subgroup of apparently healthy participants of Bauhaus. So we are doing this in a way to complement the deep clinical phenotyping and the genotyping with the metabolite data. So we are acquiring the data on a very wide diversity of polar metabolites, steroids, including sex steroid hormones and corticosteroids, and then all of these complex lipids, some of which I have shown you today. And this with this main three aims, I would say, the first one being to really define or establish the reference values and intervals for most of these metabolites that would be relevant for Swiss population. Then, in order to really, let's say, enhance, improve our knowledge about the mechanism of action of these metabolites, so their really role in disease onset and development, and then finally, hopefully, to significantly really improve this risk prediction models that could hopefully be really introduced in clinics to extend our health span. I think this is becoming quite trendy today, and we have worked a lot actually with the group of Professor Arno Schmidt-Rückes in Basel. So we have performed one lipidome-wide association study on the complete health cohort. And for this preliminary study, so consider these results are really preliminary because at the moment we have profiled quite a small sample sizes, except in Colaus. But here, for example, we have selected two different, two contrasting populations within the complete health. So the young participants in their 20s, around 70 participants in their 20s, and around 70 participants in their 70s. So please consider that they were all went through an extensive clinical characterization, and they were characterized as clinically healthy and physically active as well. So this was done with this, the measurements were done with the accelerometer, and I know that they satisfy this World Health Organization recommendations of, I think, more than 150 minutes of activity per day. Me too, I'm wondering, it seems a lot to me, but maybe you can explain me better how this was really done. But so we actually collected, so the serum was collected from these participants, and we have done the lipid profiling, so really the global omic scale lipid analysis. This was done with untargeted already 2 years ago, when we still, at that time, we still did not have yet we had not yet set up this high coverage targeted methods. So this is just to show you the clinical characteristics of this population. I mean, to prove you in a way that, I mean, what were the parameters that, what were these average values of BMI, of total lipids, and physical activity, so that they fulfilled in a way this, let's say, criteria to be classified as clinically healthy and physically active. So our main aim in this study was really to focus, because we had healthy participants, to focus on sex and age as the, I mean, key variables that will really determine, I mean, that a priori are known to determine really the metabolic profiles of different biofluids. And indeed, using unsupervised analysis, we could nicely, I mean, we could nicely identify. So the lipid signatures allowed us to really allow this sample clustering in aged and young. And indeed, we could observe that more than 80% of identified lipid species were actually significantly higher in age compared to young participants. And as you can see on the right side on this rain plot, which actually presents the results of the multivariable regression analysis, we could identify that these changes were mainly distributed across these classes. So mainly phospholipids, different classes of ether-linked phospholipids, lysophospholipid, and then also the sphingolipids. So we will go in more detail. So we, of course, did not, this is just a resume of the results. We wanted to explore the results in more details at the molecular species level. And here we observed that more than 120 different lipid species were significantly and positively associated with age in females and males confounded. But on the top of this, that actually these differences, these age-related differences in lipid levels were also sex-dependent. And here we could actually nicely observe that there were many distinct species belonging, especially to two specific classes, so the class of ether phospholipids and lysophospholipids, which were positively associated with age in females only. And this was quite interesting, and these results were in accordance with the results from several other bigger population studies that were not healthy though. And this specifically the AUSDIAB, so a big cohort in Australia, which was established to study really the diabetes. Please consider that these species are not necessarily deleterious in any way, because there are many actually lipid species that will be actually beneficial for the cardiovascular health. And this is especially the case of these species that were found to be higher in females compared to males. And this is something that was quite interesting. So there are several hypotheses why these enterophospholipids specifically get higher actually in females with aging. And one of them is that this de novo synthesis of this species in the peroxisome with aging is actually differentially regulated in females and males. There is, of course, a doubt that this might be sex hormone driven, but this remains to be explained. On the other side, for the lysophospholipids, there are already some reports about significantly different activity of phospholipid metabolic enzyme, and this specifically the cholesterol acetyltransferase, and then the phospholipase that apparently is indeed sex specific, and that is in a way higher, its activity is higher in females, which makes then lysophospholipids to accumulate with age. But these are just hypotheses at the moment. Please consider this has not been studied in more details. I mean, it starts now to be studied because people have somehow, we have raised our awareness that sex has been until now, unfortunately, an overlooked variable, and that it should be really considered separately in biomedical and clinical research. On the top of this, so as I said, age-related differences that were sex-dependent, by default, regardless of age, the serum lipid signature is sex-specific as well. And we could see this in young and in aged population. And on this circular plot, you can nicely see that, for example, the 3-acylglycerols will always be lower in women, no matter regardless of age. And in the same way, the sphingomyelins and some other sphingolipids will actually be higher in women regardless of age. And this apparently for sphingomyelins is true already in newborn children, and is being now really more and more explored. So we are now going forward with this. With the study of the entire complete health. So we haven't analyzed only these two contrasting groups, so around 150 individuals, and we are now including the entire cohort, which is more than 500 individuals. And on the top of this, also the complete heart cohort, which includes the participants with confirmed coronary artery disease. And so this mainly with the idea to explore, I mean, we have at the moment focused mainly on the sphingolipid metabolism. So these profiles that I have, these detailed profiles of sphingolipids that I have shown you previously. And we are trying to associate really these sphingolipid signatures with the cardiorespiratory fitness and the coronary artery disease. So we are looking for the potential associations. And this all, which of course this idea, as I said, to in a way ultimately improve the risk prediction modeling. And now I will show you some of these very preliminary results from the call-outs. So we managed to perform this analysis in the context of a master project actually during the last year. And this was done in collaboration with the Professor Kutalik team and also the Professor Marquez Vidal, so Titre. In call-outs, as you may know, they are it's really prospective population studies, so we have multiple follow-ups. I think today we are already at the 4th follow-up. So please consider that we had to focus on we have the samples from only and the data from only two follow-ups because there were no more samples available from the baseline. So we actually had to consider the first follow-up as our baseline, and then use the other two follow-ups for which the samples and the data are already available. So you can see that we tried to select the apparently healthy population. This selection was based on this exclusion criteria. So the exclusion criteria included actually the no confirmed disease, no dyslipidemia, no hypertension, so no confirmed diseases, obesity, type 2 diabetes, and the cardiovascular disease as well. Yeah, it's not, you cannot see it here. Then we have also discarded women who were on hormonal therapy, because we do know that the hormonal therapy can significantly influence actually the metabolic profile, and also the women who had the polycystic ovary syndrome. We have, in the context of this master project, actually, we managed to acquire the plasma amino acid and acyl carnitine signature that englobes more than 600 different species from this healthy population at the baseline. So altogether, more than 2,600 participants. And the data are, of course, available beyond amino acid and acyl carnitines. But what we managed to analyze so far mainly focuses on these metabolites that are heavily really involved in energy metabolism. And here you have the first results. So first we performed just the simple unsupervised analysis and the cross-sectional association study to look at the sex differences. So we could nicely see that only even those amino acid and acyl carnitine signatures have allowed us to cluster females and males separately. And using the logistic regression analysis, univariable in this case, we could nicely observe the significant differences actually between males and females. So globally, most of the amino acids were present in significantly higher concentrations in male plasma, with the exception of these 4 different species, including the creatine and glycine. And there are many which were really considered as the most significant differences. And there are many plausible explanations for this. And the hypothesis actually that the creatine metabolism is really regulated by the estrogens. And we know that, yes, indeed, the creatine synthesis is driven or modulated at least by the estrogens, including its transport and the creatine kinase expression. But yet another, hypothesis that actually builds up on the previous one is clearly the higher muscle mass and physical activity in men. That this is why probably we see most of the amino acids significantly higher in male plasma compared to female. But specifically for creatine, there's this hypothesis that then actually the creatine it has a higher bioavailability in females, because in a way it's less used, so it's less frequently actually transformed or degraded to the creatine compared to. On the top of this, I mentioned glycine, and glycine, this is one very important difference, because the genetic determinants behind this circulatory levels of glycine have been revealed. So we do know that there is this sex-specific or the female-specific association of the carbamoyl phosphate synthase with the circulating glycine levels and the lower risk of developing coronary artery disease. So the carbamoyl phosphate synthase actually will catalyze the ammonia, will make the conversion of ammonia to carbamoyl phosphate that will feed into the urea cycle. And it is believed that in females actually it has been proven through this through the quite big, very large-scale GWAS studies, that the activity of this enzyme is lower in females, which then makes actually which as a consequence has this accumulation of glycine and higher levels of glycine in plasma compared to males. But what is really interesting is that these higher levels of glycine have actually been associated with the lower risk of coronary artery disease, and not hormonal. So there is this belief indeed that there is a sex-biased regulation actually of ammonia metabolism via the hepatic urea cycle. And I have cited below these GIVA studies, which nicely demonstrate this really sex-specific association, so really female-specific association that of regulation glycine levels. On the top of the amino acids, we have also seen a very significant changes in the acylcarnitin profile, and most of the acylcarnitins, except for the adipoyl carnitin that unfortunately you cannot see here because I put, oops, sorry. Well, you will have to believe me, or I can show you later on. Except for the adipoyl carnitin, all the other species were indeed present in significantly higher concentrations in males compared to females. And there are hypotheses that this is related to the differences, as I mentioned earlier, in muscle mass and metabolism, so really the energy utilization in different physiological states in females and males. And also, of course, the potential implication of estrogens and adipokines that are present in higher levels in women on muscle metabolism and specifically mitochondrial metabolism and the beta oxidation. Because acylcarnitins are really markers of the, in a way, incomplete beta oxidation, so some perturbations with the fatty acid oxidation. Finally, we followed up by the longitudinal association analysis, because despite of the fact that we had the metabolic data acquired only on the baseline, we had all the other data so related to many of these cardiometabolic risk factors for this population at different follow-ups. And this allowed us actually to evaluate how the metabolites would actually, whether the metabolites play an important role on the changes, or whether they affect actually the changes in different cardiometabolic risk factors. And here we have focused mainly on the adiposity markers, so the changes in BMI and waist over time, so waist circumference, the changes in the blood pressure, so diastolic and systolic blood pressure, different diabetes markers, so the glucose levels, and the approximate for insulin resistance. Finally, we also looked at the effect on menopause, I mean, how this metabolic profile changes with the menopause. So in collaboration with Professor Kutalik's team, we actually applied 2 different models to test for this association between metabolic levels and the changes in cardiometabolic risk factors. And we have considered only the metabolites that showed really the significant association in both of these models, and for which the direction of association was the same actually coming out from both of the models. So as you can see, the second model is actually normalized for the changes with age, whereas in the first one, we used always the levels of metabolites at baseline and the first follow-up. So we had to adjust for these levels to actually calculate the potential association with the changes at the follow-up too. So finally here, some results. So we have seen a very significant positive association between the levels, concentrations of glutamate and the changes in the waist circumference, which is considered as one of the important adiposity markers, and also between the creatine and the changes in the BMI. What is quite interesting is that this first association between the glutamate levels and the waist circumference was really sex-specific, so it was found to be significant in males. So I will skip some just not to keep you too long, but we can discuss. Then when it comes to the markers of diabetes, we found many positive associations that were in accordance with the findings from the literature. So we found that the several different branch chain amino acids And on the top of them, aspartate, creatine, and amino adipate were positively associated with the changes in glucose level and insulin resistance. Interestingly, this again, this association between the creatine levels and the HOMA, so the model of insulin resistance, was again sex-specific and was significant in males. And importantly, on the other side, we could nicely confirm that yes, the glycine was actually negatively associated with the changes in glucose level and increasing glucose levels and the insulin resistance, which was, of course, in accordance with what I showed you previously, that glycine levels were actually associated with lower risk of coronary artery disease and insulin sensitivity and not the resistance. Finally, we have seen many changes also with the menopause that remain to be explained, because there is almost no studies have been performed on these changes. So there may be two or three papers that report some changes, but on a very, very small panel of metabolites. So we could see really a multitude of acylcarnitine species that were found to decrease actually with this transition to menopause. We found several amino acids, specifically those implicated in the urea cycle, that also showed a decrease with the transition to menopause, and several others were showed rather the positive, the positive association or the increase with menopause. But like I said, indeed, there, we have to do further research to really better understand these changes and how they relate to energy metabolism. So for sure, probably they do relate to the fatty acid oxidation and to the amino acid metabolism. But yeah, this has to be we have to perform further studies to get really the idea, and we have to also accurately characterize the hormonal status. So we went a bit further, and we wanted to explore the causality actually of these revealed associations. And here we have actually used the Mendelian randomization technique. And this was done based on the summary statistics available from different genome-wide association studies. And through these summary statistics, we could actually identify the levels of which metabolites had the determinants, genetic determinants. So which SNPs, actually, which single nucleotide polymorphisms or genetic variants were actually significantly associated with which metabolic levels. On the other side, we used the summary statistics from the UK Biobank also to identify which SNPs were also associated with this complex metabolic traits. So mainly these 7 cardiometabolic risk factors that we have selected to study in Colloque. And finally, using using Mendelian randomization, we could identify 40 out of 60 metabolites that were indeed associated to one of these 7 complex metabolic traits, but only 2 of them actually came and could be confirmed when we considered this significant causal effect that was determined through Mendelian randomization analysis, and then also on the top of it, the significant regression coefficients that we got out of the longitudinal models. I think. And so, yeah, we could really confirm this significant causal effect of creatine on BMI, and then the glycine on insulin resistance, which was, like I said, in accordance with this glycine lowering effect on insulin resistance. So yeah, now it's just really to finish the perspectives. We got the SNF grant to continue with the studies of the sex hormonal status and sex differences. So we will really characterize the hormonal status accurately using the steroid profile in the entire Colloque population. And then we will try to evaluate, actually assess this causal relationship between this hormonal status, the circulatory lipid signature, and the cardiometabolic risk factors. So this is really to understand to which extent this hormonal transition actually influences the lipid metabolism and then also the predisposition to develop different cardiometabolic diseases. And then, of course, the last objective is more related to really the improvement of risk prediction equations. So yeah, with this, I just want to say special thanks to the entire Metabolomics team. So we are now a team of around 10 people. It has significantly evolved since 2015 when we were established. We have 6 different instruments, so this also has significantly evolved. We do keep attention, keep taking care of our physical activity, not only doing a lot of serious lab work. And then I would like to, yes, many thanks to the Complete Health and the Colloque team, of course. So the Professor Schmidt-Trückes and Justin Carrett, who really with whom we work on Complete Health, and then of course the Pedro, Zoltan, and Peter, with whom we are in more direct contact for the Colloque. Many thanks to all the other collaborators, including the vendors who help us, of course, with the standard solutions, instrumentation, etc., as well as the NIST Institute of Standards, which is really important because they are now developing reference materials for these complex biological matrices. And yes, thank you very much for your attention. I'm sorry to be too long.
Thank you very much. I see that it is noon already. I don't know if there is any question here in the room, or Marina, you tell me if you have a question online. Okay. Yeah, you have a question? Oh, sorry, I thought you had raised your hand. Okay. Maybe I just have one question. You mentioned about standards, because usually when those type of techniques we talk about variability across labs, the influence of pre-analytic conditions. Maybe could you comment on that, on what you produced on Colloque biosamples? Would another lab produce the same results as you did?
Indeed, there was a big doubt about this, and this is why there are now strategies that have been established to harmonize the data. We are involved in different interlaboratory trials where we work specifically on these type of strategies, and one of them are reference materials. So the idea is now that every time when we perform this quantitative analysis, we do analyze a reference material that is commercially available. So there is this standard reference material for plasma that was produced by NIST in US. And so every time we analyze sample batch, we always analyze as well this reference sample. And then thanks to this reference sample, we can actually normalize the concentrations that indeed, yes, there can be significant variations depending on the methodology that was used, especially when the sample preparation is different and use different methods to measure the metabolites. But yes, with the internal standard spike and the reference materials, indeed, we can nicely harmonize the data. There are many studies that show this. We can get down to the same ranges of values, definitely.
Thank you very much. So thank you again. Thank you very much.